So, picture the mitochondria in your cells. Like, if you're relying on your old high school biology textbook, you're probably picturing this, static, jelly bean-shaped capsule.
Right, with that little squiggly line drawn back and forth inside it.
Exactly, the classic powerhouse of the cell. Well, it turns out that image is a complete lie, or at least, it's woefully incomplete.
It really is. It's a huge oversimplification.
Yeah, so today we are dismantling that textbook jelly bean. We're doing a deep dive based on this massive, comprehensive 2026 review article from Cell Death and Disease.
That is a fascinating read.
It really is. Our mission today is to explore how those inner squiggly folds, which are called the cristae, are actually these highly dynamic, shape-shifting machines.
Yeah, they aren't just squiggles. They are active structural barriers.
Right, and they dictate cellular life, cell death, and bizarrely enough, even your systemic body weight.
To really grasp what's happening inside you right now, we need to totally shift how we visualize the cell.
Okay, let's unpack this. How should we be picturing it?
Imagine a microscopic landscape, almost like a molecular star chart. Mitochondria are essentially these bustling cities within your cells.
Wow, okay. Bustling cities.
Yeah, and the fundamental unbreakable rule of these cities is that form equals function. The architecture of these microscopic folds isn't just like a container for chemical reactions.
The architecture is the reaction itself.
Exactly. But to understand why it's built this way, we really have to look back at its ancient evolutionary origins.
I want to spend a second on that because the origin story here is just wild. Before we can even talk about how these structures power your heart or your brain, we have to look at where this crazy shape came from.
Right, we're talking about the endosymbiotic theory.
Yeah. So think back like a couple billion years to the primordial soup.
Very long time ago.
Right. Our early cellular ancestors, which were likely single-celled archaea, essentially swallowed a free-living, oxygen-breathing bacterium.
But instead of digesting it for a quick meal, they actually struck this permanent evolutionary deal.
It's crazy to think about. The host cell provided shelter and nutrients, and the bacterium provided this, massive upgrade in energy production.
And over millions of years, that bacterium evolved into the mitochondria we have today.
And the craziest part is those inner folds, the cristae. They deeply resemble actual structures found in ancient bacteria called mesosomes.
Right. Mesosomes are basically these inward folds of the bacterial membrane. They help the bacteria survive stress.
Because it instantly increases their surface area, right? So they can pump out more energy.
Exactly. And when our cellular ancestors adopted this bacterium, they didn't erase that bacterial blueprint. They kept it, and they just built upon it.
So they kept the original floor plan.
Yeah, and the absolute proof of that ancient history is in the building materials themselves. If you look at the inner mitochondrial membrane, the IMM, it is completely alien compared to the rest of your cell membranes.
Oh, wow. Alien how?
Well, first of all, it contains absolutely zero cholesterol.
Wait, none?
None. Instead, it is packed with a very unique ancient lipid called cardiolipin, which makes up about 15 to 20% of that inner membrane.
Okay, so cardiolipin is basically the master architect of this entire city.
It really is.
I love the mechanics of this, by the way, because most lipids in your cell membranes, they're somewhat cylindrical,
Yeah.
They have two tails, and they pack together nicely to form flat sheets.
Like the boundary wall of a normal cell.
Right. But cardiolipin has a totally unique conical shape. It has 4 of these fatty acyl tails instead of the usual 2.
Which is a huge structural difference.
Yeah. Because it's shaped like a wedge or a cone, when you pack a bunch of them together side by side, they physically force the membrane to curve inward,
creating this incredibly tight fold.
It totally reminds me of traditional stonemasonry. Like if you're building a stone archway, you can't use standard square bricks at the top of the arch.
You'd never get the curve.
Exactly. You have to use keystone bricks, those wedge-shaped stones that allow the structure to curve sharply without, collapsing under its own weight.
And cardiolipin is exactly that keystone. But it's important to note, it doesn't just spontaneously form the perfect wedge all on its own.
Oh, really? It needs help.
Yeah, the membrane requires a cellular renovator. There's this vital enzyme called tafazin.
Tafazin, okay.
Right? When cardiolipin is first manufactured by the cell, it has these generic, bulky lipid chains. So tafazin comes in and systematically swaps those generic chains out for very specific ones.
Basically upgrading the material.
Exactly. Predominantly swapping them for something called linoleic acid.
Right.
This active remodeling process is crucial because it significantly reduces the biophysical tension of that extreme curve.
Got it. So it allows the membrane to handle an incredibly dense packing of proteins without just tearing apart under the pressure.
You hit the nail on the head.
But I'm trying to visualize this in a living, breathing cell, though. Because if cardiolipin is just a structural brick or a keystone, how can these folds be so dynamic?
What do you mean?
Well, the review highlights that cristae change shape incredibly fast based on energy demand. Like when one of your immune system's T cells suddenly gets activated to fight off a virus, its mitochondria instantly shift.
Right, from a relaxed, wide-open orthodox state into a super tight, condensed state.
Yeah. And Brits don't just spontaneously reshape themselves in a matter of seconds?
No, they don't. Which is why we have to view cardiolipin not as a rigid brick, but as a highly active fluid scaffold.
Ah, okay. A fluid scaffold.
Right. It basically acts as a dynamic shock absorber. When your cell's energy demands suddenly spike, like with that T cell fighting an infection, cardiolipin allows the membrane to flex and compress.
So it instantly accommodates the physical crowding of like thousands of new proteins rushing in to produce energy.
Exactly. It maintains that extreme hairpin curvature without the membrane snapping.
That is wild. Okay, let's do a quick mental checkpoint for everyone listening because we're building a really complex machine here.
Good idea.
So far, we've got this ancient bacterial membrane folded inward, acting as our foundation. We've got cardiolipin acting as the wedge-shaped keystone, making the curve possible.
Right.
And we've got the enzyme tafazin doing the remodeling to keep the tension manageable. But, I mean, that's just the foundation. Who is actually holding the doors open and keeping the roof from caving in?
Well, that brings us to the construction crew. You have three main players actively managing this architecture.
Okay, who are they?
You've got the MICOS complex, OPA1, and ATP synthase. Let's start right at the entrance to the fold.
Right at the doorway.
Yeah. The MICOS complex acts as the static anchor at what we call the cristae junction. Imagine a balloon, right? The cristae junction is the narrow neck where you tie it off.
Oh, that makes sense. So these junctions are incredibly narrow bottlenecks connecting the deep folds to the outer boundary membrane.
Precisely. MICOS is the physical scaffolding that stabilizes that bottleneck. It ensures the inside of the cristae remains a completely distinct, quarantined environment for the rest of the mitochondrion.
Quarantine, right. And working hand in hand with MICOS is our dynamic remodeler, right? A protein called OP1.
Yes, OP1 is crucial here.
I always picture OP1 as this incredibly strict bouncer standing right at the nightclub door of the cristae junction.
That's a great way to look at it.
Yeah, these OP1 proteins link up with each other to form a physical staple across the opening. By tightening or loosening that staple, OP1 mechanically opens or chokes off that bottleneck.
It literally reaches out, grabs the cardiolipin chains we talked about earlier, and shifts them around.
Oh, so it physically interacts with the cardiolipin.
It does. It uses them to decide what molecules get to leave the fold and what stays trapped inside.
That is a masterclass in dynamic control.
It really is. But if OP1 is guarding the door, we have to look deep inside the fold at the very apex of the curve to find ATP synthase.
The famous ATP synthase. Usually biology classes just describe it as the turbine, right? The molecular motor that spins to create ATP, which is the energy currency your body uses to do everything.
Right, but it actually has a crucial second job that gets overlooked.
Okay, what is it?
These ATP synthase enzymes assemble into V-shaped pairs, or dimers, and they sit exactly at the sharpest points of the cristae tips.
Like little tent poles.
Yes, they act exactly like molecular tent poles pushing up against the membrane. If a cell loses these tent poles, the cristae lose their sharp tips completely.
And they just collapse into these useless, abnormal, onion-like structures.
Exactly.
I'm a little confused, though. If ATP synthase is the engine making the energy, why is it also doing the heavy lifting of physically pushing and shaping the membrane?
It seems like double duty, right?
Yeah. Like, why burden the engine with structural engineering? Isn't that doing two jobs at once?
What's fascinating here is that this is one of the most brilliant evolutionary adaptations in our biology. It comes down to a concept called kinetic coupling.
Kinetic coupling. Okay, what does that mean?
By physically shaping the membrane into these tight, highly curved tips, ATP synthase is actually manipulating its own immediate environment to be more efficient.
Oh, really? How?
It creates what we call a localized proton trap.
A proton trap. Wait, explain that.
So in order to spin and create energy, ATP synthase needs a constant flow of protons, basically like water flowing over a water wheel.
Right.
Because it forces the membrane into such a sharp curve, the physical space right at the tip is incredibly confined.
Oh, I see.
This creates a massively steep pH gradient right at the exact location of the ATP synthase motor.
Wow. So it essentially traps the protons and funnels them directly into its own turbine rather than letting them float away into the wider cellular space.
Exactly. It is literal structural engineering designed to guarantee maximum energy output.
That is absolutely brilliant. It's physically folding the membrane to funnel the fuel directly into its own mouth.
It really is.
And what regulates this whole operation, I know the review mentions a molecule called NAD plus Dale.
Yeah, NAD plus is basically the fuel gauge and redox sensor for the whole system.
Okay.
When your cells are in a low energy state, NAD plus levels rise. This activates a specific family of anti-aging proteins called sirtuins.
We'll take this RT3, right?
Exactly. Specifically RT3 in the mitochondria. RT3 then chemically modifies our nightclub bouncer, OP1.
Oh, so it keeps the bouncer active.
Right, ensuring that crystal junctions stay tight and efficient. It's an elegant feedback loop. The metabolic state of the cell dictates the physical architecture, and the architecture dictates the metabolic output.
That is so cool. But building this elaborate proton trap is great for energy, sure. But running an engine that hot creates a massive side effect, doesn't it?
It does. Toxic exhaust.
Right. The electron transport chain, which sits in these deep folds, generates highly reactive, dangerous molecules called reactive oxygen species, or ROS.
And ROS is very destructive.
Exactly. And if we think back to our origin story, mitochondria kept their own DNA, mtDNA, from their bacterial days.
They did.
So how does the cell stop that toxic exhaust from just mutating and destroying its own genome?
Well, it solves the problem through strict compartmentalization. The physical architecture acts as a literal shield.
How does it shield it, though?
The mitochondria deliberately anchor their DNA out at the boundary membrane, as far away as possible from the deep cristae tips where the toxic ROS is being generated.
Oh, wow. So by structurally keeping the DNA in the shallow end, basically, and the toxic exhaust in the deep end, it protects its own genetic blueprint from oxidative damage.
Exactly. It's separating the hazardous waste from the control room.
That makes total sense. And this idea of compartmentalization gets even more extreme when you look at the cell as a whole. You and I were looking at this groundbreaking 2024 study by Ryu and colleagues.
Oh, yeah. That study is phenomenal.
It fundamentally changes how we view these organelles.
It really does.
Yeah.
Because for decades, we just assumed that all mitochondria within a single cell were generally doing the exact same job.
Just making ATP.
Right. But Ryu's team discovered that a single cell can harbor two entirely different, structurally distinct populations of mitochondria operating simultaneously.
Wait. In the exact same cell?
In the exact same cell.
That is insane.
They found that some mitochondria lack cristae entirely. They have absolutely no folds.
Okay, so what do they do if they don't have folds?
Well, because they lack this specific architecture, they cannot perform the normal energy-producing reactions.
Right, no proton trap.
Exactly. Instead, these cristaeless mitochondria are entirely dedicated to a completely different chemical process called reductive biosynthesis.
Reductive biosynthesis. Got it.
They rely on an enzyme called P5CS to synthesize vital amino acids like proline and ornithine.
Wow. Meanwhile, the mitochondria sitting right next door in the same cell are just packed with tight cristae and are solely focused on churning out ATP.
Exactly. The physical presence or absence of the cristae acts as a literal structural switch to separate competing chemical reactions.
That completely shatters the jelly bean myth. Like, the mitochondrion isn't just a bag of chemicals. It is a highly zoned, adaptable manufacturing plant.
It absolutely is.
But so, what does this all mean when things go wrong in that plant? Let's say one of those folds gets hit with too much of that toxic ROS exhaust and just takes irreversible damage.
Well, this is where we see their ultimate survival mechanism. They use something called VDIMs.
VDIMs. That stands for vesicles derived from the inner membrane, right?
Yes. So imagine a large submarine taking heavy fire. If one compartment floods, you don't let the whole submarine sink.
No, you seal the heavy bulkheads.
Right. You isolate the flooded compartment and keep the rest of the ship functioning.
And the mitochondrion does the exact same thing. The damaged cristafold literally pinches itself off from the rest of the inner membrane.
It structurally isolates itself.
Into this tiny free-floating bubble, a VDIM, and then that little package of damaged goods is exported out of the mitochondria and sent to a cellular recycling center.
Called the lysosome, yes, to be destroyed.
So the mitochondrion just amputates the bad part to save the whole. It basically performs surgery on itself.
It really does.
But wait, if they are constantly amputating parts of their own inner membrane and throwing it in the trash, how do they not just run out of membrane and shrink to nothing?
That's a great question. It's because mitochondria are never truly solitary jelly beans. They exist in these massive dynamic networks that are constantly fusing together and dividing apart.
Ah, okay.
This fusion and fission cycle is continuously mixing healthy membrane lipids and proteins across the network.
So it's constantly replenishing the lost building material.
Exactly. It's a perfect continuous balance of quality control and rebuilding.
That is just a masterclass in biological engineering.
Yeah.
Let's do another quick mental check. We've seen how they use ancient bacterial blueprints to build a proton trap.
Right.
We've seen how they use structural switches to separate competing chemical reactions. And we've seen how they amputate their own damaged parts to survive.
It's incredible.
The architecture is flawless until it isn't.
Right. What actually happens to you, the human being listening to this, when a single protein or a single lipid in this microscopic fold malfunctions? Well, the consequences are catastrophic, and they manifest across a huge spectrum of severe human diseases.
Because form equals function. Right.
Exactly. When the architecture collapses, the organ starves. Let's look at neurodegenerative diseases first, like ALS.
Okay, KLS.
Your neurons have massive constant energy demands just to keep your brain functioning and your muscles moving.
Right. They need tons of ATP.
Well, in ALS, pathological proteins called TDP-43 begin to accumulate directly inside the mitochondria.
Oh, no.
And these toxic proteins physically destroy the cristae structure. Without the folds, the proton trap vanishes, ATP production plummets, and the neurons literally starve to death.
Which leads to the progressive paralysis we see in ALS patients. That is heartbreaking.
It is. And it's not just the brain. It impacts the heart just as violently.
Right, like in cardiovascular diseases, right, like hypertrophic cardiomyopathy, where the heart muscle thickens and struggles to pump blood.
Exactly. When researchers look closely at those cells, the cristae inside those heart cells have become wildly disorganized.
But the review highlighted something really incredible here. There is this old drug called pyridostigmine, which has been used for decades to treat a completely different muscle disease called myasthenia gravis.
Yes, and it turns out this drug can actually reverse this heart failure.
Wait, really? How?
By upregulating OPA1.
Our nightclub bouncer.
Exactly. It sends more of our nightclub bouncers to the cristae junctions, retightening the bottlenecks and restoring the architecture.
So if you can fix the microscopic structure, you can actually restore the pumping function of a human heart.
That's the power of this architecture. And unfortunately, we see the tragic inverse of this in a genetic condition called Barth syndrome.
Barth syndrome. Yeah.
This disease is caused by a tiny mutation in the TAZ gene.
The TAZ gene. Wait, that's the blueprint that creates tafazin, right? Our cellular renovator from earlier.
Yes.
The enzyme that remodels cardiolipin so it can hold the curve.
Precisely. Without a functioning tafazin enzyme, the cardiolipin lipids are never remodeled. They keep their generic bulky shape.
Oh, man. So because they are the wrong shape, they cannot hold that extreme and negative curve.
Right. The entire lipid foundation is faulty. And because the foundation is unstable, OPA1 detaches from the junction, the ATP synthase tent poles fall apart, and the entire crista physically collapses inward.
That's devastating. And this microscopic collapse leads to severe, often fatal, heart failure and metabolic crises in young boys, all because a single type of lipid couldn't be shaped correctly.
It is incredibly sobering to think that human life hangs on the structural integrity of a single lipid.
Here's where it gets really interesting, though. We've talked about how this impacts the brain and the heart, which are obviously high-energy organs. But what about the waistline?
Yes.
The review delves deeply into how cristae architecture directly controls systemic obesity. I mean, how does a microscopic fold in my cell affect my body weight?
If we connect this to the bigger picture, it is one of the most fascinating systemic connections in modern biology.
Okay, lay it on me.
Deep in the brain's hypothalamus, you have a specific cluster of cells called POMC neurons.
POMC neurons, got it.
One of their primary jobs is to monitor your body's energy levels, suppress your appetite, and send signals to your adipose tissue, your body fat, telling it to break down and burn energy.
Right, they're essentially the metabolic command center.
Exactly. So researchers decided to run an experiment. They genetically deleted OPA1, just the OPA1 bouncer protein, and they only deleted it in those specific POMC brain cells.
Okay, just in those cells, what happened?
The cristae junctions in those specific neurons dilated. They lost their tight bottlenecks.
And what is the crucial how and why of that? Like, why does a dilated bottleneck cause obesity?
Well, when those cristae dilate and the structure loosens, the mitochondria lose their ability to properly buffer and regulate calcium levels inside the cell.
Wait, calcium. I thought calcium was just for bones.
It's not just for bones. In neurons, calcium spikes are the exact trigger required for the cell to release communication messages.
Oh.
Because the mitochondria couldn't regulate the calcium, the P, O, and C neurons failed to release a specific hormone called alpha MSH.
And alpha MSH is the messenger hormone that travels out to your body and tells your fat cells to undergo lipolysis, which is the breakdown of fat.
Exactly.
So just to be totally clear, because a microscopic bottleneck loosened in a single cluster of brain cells, the calcium levels flatlined, the alpha MSH hormone vanished, and the body literally forgot how to break down fat.
That is exactly what happened. The mice in the study experienced massive systemic weight gain and severe obesity, all from a microscopic fold dilating in the brain.
That perfectly bridges the micro and the macro. It reveals that mitochondrial microarchitecture isn't just about, local energy production. It is inextricably linked to systemic whole body metabolic signaling.
Right. A structural change measured in nanometers dictates a physiological change measured in kilograms.
These ancient folds aren't just engines. They are the master control switches for our entire metabolism.
They absolutely are.
This has been an absolute journey. I mean, we started by looking at an ancient bacterial survival trick, the mesosome floating in a primordial soup. And we've ended up with this highly regulated, shape-shifting architecture that controls everything from our beating hearts to our expanding waistlines.
It's pretty amazing.
We are looking at a dynamic, intelligent machine that uses keystone lipids, molecular bouncers, and kinetic proton traps to keep us alive. It structurally quarantines its own DNA, changes its shape on demand, and can even amputate its own damaged parts to survive.
It's the ultimate survival machine.
So I want to turn this over to you, listening right now. What stands out to you? Is it the fact that your cellular engines can essentially perform localized surgery on themselves to quarantine damage?
Or maybe it's the mind-bending reality that your body's ability to burn fat relies entirely on the architectural integrity of a microscopic bottleneck in your brain.
Those are both profound mechanisms to reflect on.
They are.
Okay.
But I'll leave you with a final, perhaps slightly unsettling question to ponder as you go about your day.
Oh, boy.
If our mitochondrial cristae are fundamentally built on ancient, alien bacterial blueprints, and if those microscopic structures possess the absolute power to dictate our cell death, control our immune system responses, and even regulate our systemic body weight, are we truly the ones driving our complex human biology?
Oh, wow.
Or are these ancient microscopic guests still secretly pulling the evolutionary strings?
That is a wild, lingering thought to end on. If you want to dive even deeper into the structural engineering of your own cells, check out the 2026 Cell Death and Disease Review we explored today. Thanks for joining us on this deep dive. We'll catch you next time.